Thanks, Bernd. Yeah, so my sort of thing that I've been dealing with a lot is my work often crosses from taxonomy to conservation. And that means that there's a lot of challenges in biallelic data that need to be really clearly addressed when we are and thought about when you're designing your study. And so today I'm going to talk about why study design is so important and why how you call SNPs is really fundamental to the actual results you get out of your data. And try and give some clear examples, and then point you in some pathways that sort of lower the challenge threshold for being able to do this yourself. So ddRAD versus whole genome sequencing is a subset of the genome. So you know that it's cut by common and rare restriction enzymes, and then those genomic fragments that we've selected that are based on those, they're size selected, and then they get barcodes on them and then amplified. And once we have all of that data, those reads are assembled into loci. And on those loci that we have selected, we have discovery of SNPs being biallelic sites during that assembly. And so during this process, a lot of things can bias the data that you get out of the process. So not all loci are recovered in all individuals because of variation in sequencing depth, the restriction site presence. So if you have loci, if you have multiple species in your data set, that changes your loci significantly because you get locus dropout through mutations within the restriction enzyme binding sites. And so that can fundamentally change what you get out. And so missing data, locus dropout, common features of data sets like this, and enormous number of factors and settings affected. And I wish I could tell you do these 5 things and it will be fine, but the answer is everybody's data set is kind of different. Everybody's species is different. Everyone's question on the same species can be different. And all of these things mean your SNP catalog can vary. Your catalog of loci varies because of the individuals, the question, the locus discovery, and the filtering that you do. And so it's not a fixed set of genomic sites. It emerges from the data that you use to construct it. And what we're really talking about is ascertainment bias. So ascertainment bias is a systematic deviation, giving you a really specific definition, the systematic deviation of population genetic statistics from theoretical expectations. And that can happen because of non-random sampling, small sample sizes, imbalanced sample sizes, and then biased protocols when you're actually discovering your SNPs. And so this is, I'm going to talk mostly about stacks. It's what I use mostly because it's pretty well documented. Mentioned later, I don't have an issue with any particular SNP discovery bioinformatic pipeline, but I use this because it's easy to teach and it's well documented, and in Galaxy, which means the knowledge for entry is much lower. So the first stage is locus discovery, and that's this section right here, which is where you have all of your reads for locus, and they're built into stacks, which are the reads representing the same allele. And then these are assembled into a catalog of loci. So this is where they're getting aligned across all of the individuals. So you match those loci to identify which ones are homologous, so of the same evolutionary origin. And then creates this catalog of loci. Once you have that catalog, it's set. And you can only discover SNPs within that set of loci. The second set is through this G-stacks and populations phase, where within the catalog of loci that you've restricted yourself to, you assign genotypes to individuals. And after that, you have SNPs, which you then filter for various purposes. So it's a really broad overview, but it's really important to remember that the decision about a locus is, there's a design strategy to that in C-stacks, and then there is a SNP discovery stage. And this is where you're restricting yourself to biallelic sites of your selected individuals in the loci that you have built in your catalog. So they're only identified within those loci discovered and retained in construction. And the catalog matters. So in RAD pipelines, the SNP discovery, as I said, occurs in that catalog, but the loci only exists if they were successfully matched during that catalog building stage. And the challenges we see is if you remove or add individuals, it changes the loci that are retained. So you can't just go and add some more individuals and find the SNPs and think, oh, there actually could be more loci that become more informative. So the consequences of this is the number of loci and SNPs change when the sample set changes. So the comparability needs to start, you have to start actually at the raw data stage. Allele frequency distributions change when you change your suite of individuals, and given population genetics pretty much entirely based on allele frequencies, that actually means you can change your answer. And so basically your SNP data set is shaped by how you design your study, and that needs prior thought. This is a good time to do the polls I wrote. Bernd, how do I do that?
Which poll, sorry?
I wrote polls. How do I put my polls?
Polls, oh, okay, sorry, yeah. Which one you have?
Okay.
And
a ton of people have voted already, but I would love it. I see it, show it on stage. If you haven't voted on this, I want to ask, how have you obtained, actually I'm going to start with this one, show this one. How did you select your individuals for SNP calling? Because this just kind of gives me a background of how people went about it. I think this is really interesting. I'm glad to see a bit of both moving up in the poll. Okay, so I used as many available individuals as I had access to and could afford is around the same as I did that, plus I tried to get equal representation. So that's not bad, and lots of people haven't done it. Okay, so in this one, how did you get them? So I called them myself. They're provided by a commercial company, or I don't have them yet. So provided by a commercial company. That's good to know. There's absolutely nothing.
Sorry, Renee, sorry to interrupt.
Yeah.
I think when you set up the poll, or it doesn't matter, they can't seem to see the results. So can you read them?
Okay. Sorry.
I don't know why, because it should be on stage. I don't get it.
I don't either. Okay. So in this, how have you obtained SNPs for your study? We've got around 13% called them themselves. 64% said their SNPs provided by a commercial company, such as Diversity Arrays or AGRF, and 22% said they don't have them yet. So that's interesting. Close that. My poll. Go back. Okay. That's fine. I'll say repeatedly, there is nothing wrong with the bioinformatic pipelines from commercial companies, as long as you as the scientist have designed the study properly and told them what you want, which is a you thing, not a them thing. So the first thing we have to talk about is that absolute sample size affects SNP discovery. So the number of individuals included in a data set influences which loci and SNPs are detected. And this can be up and down. So more individuals increase the probability of finding loci with rare polymorphisms, which are important to conservation genetics. And so we get that increase with sample size. We get improved estimation of allele frequency. We get more confident in our allele frequencies with larger N's. But what we also see is that loci have to be more present across more individuals to pass our filtering thresholds. And something that we actually have to think about is that polymorphic loci will drop out if they're not biallelic. And this can be really important when you have either taxonomic questions you're trying to get to, or perhaps you had a really widespread species that has become anthropogenically fragmented. You might actually have had such widespread species that originally, before decline, it was polymorphic. So it might have been like a loci could have gone from G to A to T across the range of the species. Those loci will be lost because they're not biallelic. So changes in your sample size do things like change the number of SNPs detected and number of polymorphisms. We see shifts in minor allele frequencies. So you get more rare alleles the bigger your sample is, and that can change things like demographic reconstructions. But again, we lose our polymorphic loci, and it can change, it does change our estimates of diversity, because when you introduce lots of new individuals, you can drive down your diversity estimates, because if you think of heterozygosity as variable sites divided by invariant sites, as you add individuals, you end up tending to add more invariant sites than variant sites, which means that you, when we talk about things like autosomal heterozygosity, which means that you're increasing your denominator faster than your numerator, and therefore just adding samples reduces your heterozygosity, and that becomes problematic. So they can asymptote. Our estimates you tend to asymptote, which looks good because you're like, I got enough samples, but can also start to decline with increased sample size. So testing the effects of sample size are really important when you're doing your SNP discovery and filtering afterwards. So this is something that's so close to submitting on Antechinus argentis, which is Queensland Sky Island marsupial. It's endangered, and it lives on 3 different mountaintops in Queensland. I was quite curious about how robust my estimates of SNP-based diversity were, partially as a lesson that I could publish, that SNP-based estimates of diversity can be problematic. And what I will show you here, so what I did was I had these 3 populations with my sampling, and I basically bootstrapped. I sampled and called SNPs across my individuals repeatedly. So each of these is a set of replicates at a sample number. So I've got like from 5 to 19 individuals and my 3 populations. And what I can show you here is this is first the total number of sites. And what you're seeing is, as there's some sort of weird effects here because of sample number, but essentially all 3 of my populations, number of sites behave similarly. So that's good. But what's really important here is that the variant sites, so the sites on which we would base our SNPs, don't behave the same across our populations. So what we can see is this population, which is Bulburin, our most important genetic population, actually goes up faster, right? So what we're seeing is as we add more individuals, we continue to add variant sites because there's so much more diversity in this population, whereas the other 2 populations plateau earlier. And this becomes problematic when you think about sample size, because if you were comparing, say, 19 individuals of Bulburin to 9 individuals of either of the other populations, you're going to really misrepresent how diverse your populations are. So this is what we have here is autosomal observed heterozygosity and autosomal expected heterozygosity. And really the key takeaway I want you to have from this is if you have different numbers of samples, you cannot compare your diversity estimates across them directly. You can't say, well, I estimated diversity from one population with 19, and I'm directly comparing it to another population with 10. Those are not comparable numbers, because you are going to get this sort of issue where you're comparing different parts of this sort of accumulation curve of diversity, and that's not okay. So balanced sampling is really important across populations. So that SNP discovery is really dominated by your largest population. I'm going to show you some examples of what goes wrong with imbalanced sample sizes. So the loci and variants, more common in the large population, are going to be retained, and you're going to lose the ones in your less well-sampled population. This is really important in conservation, because often our less well-sampled populations are the ones that are harder to get to and declining the most. And we're underestimating the diversity in them if we have really biased sample sizes, and we're not accounting for that in our interpretation. So variants that are unique to that smaller population are less likely to be detected. And then we have to remember that those global filtering steps we do, like minor allele frequencies, are driven mainly by the largest group. So if you have this really imbalanced sampling and you say, I'm going to use a minor allele frequency filter of 0.05 because everyone seems to do it in the literature, you're going to lose more SNPs that distinguish population B and retain the ones that are in population A. And so you get this biased loss of alleles in your, biased loss of loci in your sampling. So this has big effects on downstream population genetic results. So common things you can see is that observed diversity can be lower in undersampled populations if we're using SNP-based diversity metrics. We can miss our private alleles in those small populations, and we bias our estimates of things like FST, and we get some weird stuff happening in our ordination analyses. So the take-home message here is balanced sampling improves that reliability of both SNP discovery, but our allele frequency estimation and our population genetic inference. So now some examples with some less boring slides. This is some data on a very cute little frog. I love this picture. They don't usually have this weird little face, but I think this one was annoyed. So this is Euprolia daviesii, a Howard Springs toadlet. It's the only endangered frog in the Northern Territory, and it only lives in the Darwin urban growth area. And it requires a very specific habitat type called sandsheet heath, which is a habitat that I suspect should be listed as a federally listed endangered ecosystem. And so this is all work done by my research student, Sheng Yao, who's sitting in here in WA. And we went up to Darwin, and what you see, this is Darwin there. And basically this is Humpty Doo, and that's 10 kilometers. So this is the entire range of the species. It's basically all, if it's not urban, it's under quite a lot of threat of development within the Darwin region. So really all within 25 kilometers of the Darwin CBD. We made an enormous effort to try and get equal sampling across the region, across the whole species. But what you see, we did vary from 10 to about 5 samples. And this is the result of our population structure analysis if we included different individual numbers, so everybody, versus the same number of individuals per site. And when we have different individual numbers, we would normally take this to say k equals 3, although this looks like isolation by distance, whereas the same number of individuals per site gives this pattern, which I would personally interpret as k equals 1, or strong isolation by distance, which is what we see in the species. So this is same data, but just calling SNPs on everybody versus the same number of individuals per site. And so what that could result in is the management of a threatened species being targeted at discrete units versus us choosing to do a restoration of connectivity in what's an anthropogenically fragmented species. So these actions can have real consequences. This is another data set I've worked on. I'm not going to tell you what it is, but it's one of those studies where it was like as much as you can get sampling. And this is essentially the same data set, but North Sydney here. So this was within the city, but this population, I think because it's accessible, was incredibly well sampled versus all of this, which is the rest of New South Wales distribution of the species. When we went back and called SNPs, and so our most divergent clade is our overly sampled North Sydney clade. But when we went back and did equal number of samples per each population, most of our variation, which also went up, so this PC axis 1 went up, became this isolation by distance. Essentially, these are the geographic range of the species from north to south sort of thing. And then we still get this interesting pattern here of North Sydney, but what I would argue is that this is a sign of bad demographic processes. This is actually something that's gone through bottlenecks and has issues, and a population that needs support. So you can very much influence your estimations of things like isolation by distance and how distinct a group is by just having the wrong number, like having a really big sampling imbalance. So what's the mechanism by which this occurs? So these are all very fancy slides, but let's say we have Australian monsoonal tropical species. So we've got thousands of kilometers, and we have two species. One that is widespread and has isolation by distance, and another that is over in Cape York. So when we think about SNPs, we get a SNP that distinguishes between species. So that means everybody over here has A and everyone over here has a T. Okay, great. But if we want to distinguish isolation by distance, a SNP data set called on both species will keep things that go from basically T to A, but retain a T here, because this is biallelic data. We've thrown away all sites that have more than 3 alleles. And this can create some weird answers for you. The other option we have is that we have a SNP that distinguishes species, so different between the two species, and also distinguishes admixture. But this is lost from your data set because it is triallelic. So what I want to point out here is if you call SNPs once across the scenario, and then you say, well, now I want to do IBD, I want to test for isolation by distance in this species, you don't get this loci back. That loci is gone and can't be recovered. And we found that to be an issue in this data set. So this was published last year by my student Cameron Dodd. So this is on the coal tar, which even as a frog biologist, I can acknowledge is pretty adorable marsupial. And it had 3 very distinct phylogenetic clades. This was true also in other data. And the weird thing was when we had SNPs called across the whole thing, what we found was that our central green was admixing with our western blue, but not with our eastern blue. And that was because we had this happening, right? We kept the SNPs that went from that where they were the same, but we lost all these ones that would have shown isolation by distance. Those are missing from our data set. And so what Cam then did was call SNPs on our contact zones between the 3 species and was able to show that there's no admixture in these zones. Back this up by lots of morphology and other things like that. But it's an example of one where we really were going, well, that's weird. And because that broader SNP data set was called on too wide of a thing, we lost our ability to, we lost all those really a lot of our useful isolation by distance loci when we had multiple species. So what I want to say again is that there's nothing fundamentally wrong by the methods that exist to get SNPs. So doing it yourself through stacks, GATK, IPyrad, fine. None of these are a problem. Having SNPs called by diversity arrays, AGRF, fine. Done it myself, published it myself. These are all good at the fundamentals of SNP calling, but poor study design means rubbish answers. And that's not something fixable by a commercial entity, because it's not their job to design your study. It's their job to take your study design and give you the SNPs. And if you ask them for the wrong thing, you get the wrong thing, and that's the reality. So it's on you, and it's not fixable without recalling SNPs. And that's a really important takeaway. This all kind of came to me, started becoming a big thing. Every time I thought about all the biallelic nature, it gave me a math stomachache. And when you think about SNPs, you need to remember that they're biallelic, explicitly biallelic, and will always identify the biggest divide in your data sets. Therefore, study design and underlying different, it creates issues with design and changes your underlying diversity. So I'm going to tell you about this one. I want to say on this paper, every single SNP data set in this paper was called by diversity arrays. I didn't do it myself, and they were great. I had to give them, they did 4 different data sets for me for this paper, and there's nothing wrong with their pipeline. I had to get what I needed out of that SNP data set right. So this is a frog, different Euparolias, fat toadlet and northern toadlet. And in this, we have one species in blue in the Kimberley and another species in yellow that's in the Kimberley and top end. And they do generate F1 hybrids. When I first did this, I use them as an example because we called data sets in 4 different ways, all of which I used in the paper, but I used them in different ways. So the first was both of the species plus the plus some outgroups. So using that for phylogenetics. Another data set was on both species, Borealis and Crassa, which is B and C, and that data set was largely used to identify hybrids. And then I called SNPs independently on each species without hybrids involved to get information on diversity and demographics. So firstly, I think this should be really convincing to you about when we lose loci and how filtering affects it. The wider we go, the less SNPs we have, okay? Because we get allelic dropout, we get our minor allele frequency filters take out more individuals, all of this sort of stuff. So we get a lot more SNPs when we focus in. And these, I like to think of these as different questions I'm asking with my data. So when I'm asking for all of this, I'm saying, give me SNPs that distinguish between many species. I'll filter them to the ones I care about. This is, give me SNPs that distinguish these 2 species, which can be quite different, and allow me to identify hybrids, and give me SNPs that distinguish population structure within Borealis and within Crassa. So try this visualization. So these are limitations because SNPs are biallelic. So let's say we have our data here. We have 2 species with 2 populations each. Actually, let's say this is 4 species. So when we have our data set, SNPs that distinguish between all of our species are going to look something like this. We'll have one allele for this clade and another allele for that clade. We keep SNPs that look like this, where maybe the change was only in this species. So everyone's a C and this one became a T. Keep that SNP as well. But as soon as we bring in a 3rd allele, that SNP is lost, okay? And so these clades will disappear. And what I want to say here is that variant sites that are heterozygous, also in the literature have a tendency to be the ones that become polymorphic across taxa, right? So it's a third code, if it's a third codon position, where the change doesn't result in an amino acid change, you can get this sort of rolling changes, which means you end up reducing, you're reducing your ability to detect admixture. When you look at that data set, which is the two species together, where I wanted to detect hybrids, you keep your alleles that where there's only two. But if you have these sites that distinguish population structure within each species, they're lost in this two species data set. And we only get to keep that with fine scale structural information that informs things like population structure and diversity and unique private alleles when we call them separately. So they're different questions, and we should expect our data to be different and more useful for different questions because of our SNP calling. So this is just a PCA on all of those variables, and all set to kind of the same individuals in my two species. So this is when it's called with a bunch of other species. This is when it's the two species and hybrids, and then the two species separately. The main thing I want to point out is that although our structure hasn't substantially changed, our amount of variance explained goes down. And that is because we're getting a lot more interesting structure in our data. We're getting a lot more interesting information in these data sets that was lost in these. Okay, so this increase in variance is because variance explained is because we've lost a lot of like really weird stuff happening within the species, which may be of conservation importance. And I just want to say, so on SNP-based heterozygosity, so we have observed heterozygosity and expected heterozygosity at each of our three data sets. And these all change quite dramatically based on our SNP calling. So I kept everything else the same in all these pipelines, but what we see is big increases as we go, as our data sets get more focused. But observed and expected, so this is observed divided by expected, don't change in ways we totally understand. And this is kind of a key part of understanding demographics, is our relationship between observed and expected heterozygosity. And so this is really biased and can be really challenging. So SNP-based heterozygosity is, I think, going up the wayside at the moment. And this started a great paper by Thomas Schmidt in Melbourne, and he showed that he looked at SNP-based heterozygosity, observed heterozygosity versus what he calls autosomal heterozygosity, which is basically the number of biallelic sites divided by the number of total sites. And he tested for the effects of sample size, of rare allele filtering, missing data threshold, population structure. And what he found was increasing that number of individuals decreases heterozygosity in SNP-based measures, which was interesting. And these are weird graphs because they're kind of flipped over, but so he found these things. So too many individuals reduces your heterozygosity. Calling SNPs on more than one population at a time decreases heterozygosity. Again, we're losing those important within population alleles, and removing rare alleles also decreases heterozygosity. And that's because rare alleles actually carry a lot of the population level heterozygosity. So how you do your rare allele filtering is really important in demographic analyses. They found that increasing missing data reduced heterozygosity, even with what they call autosomal heterozygosity, and had a series of conclusions. So that you should report autosomal heterozygosity and not SNP-based heterozygosity. And I definitely agree that autosomal heterozygosity is, for lots of things, especially if you don't have a reference genome, what you should be reporting. And they said remove sites with any missing data, and populations should be analyzed in independent runs. All of those, so we did parts of this paper because we found that those didn't all totally agree with us. We strongly agreed that SNP-based heterozygosity is impacted by sample number. So this is from our paper. And what you can see here is on the bottom is sample size number. We have 3 species, a grasshopper, a eucalyptus, and a frog. And the color gradient is our missing data allowed. So no missing data is the darkest, and 0.07 missing data is the lightest. And then we have each of the populations for that. And what you can see is as we put more stringent missing data allowed into our filters in, so the stronger our call rate filter, and that interacted with our increase in numbers. So the worst was the Litoria rubella in the Kimberley, which is as our call rate filter went up and our number of samples went up, our heterozygosity bombed. And this is the autosomal heterozygosity. And we see that also for the eucalyptus as well, and less strongly, but a little bit for the grasshopper. And so that was SNP-based heterozygosity. And so it kind of shows you, I mean, you can't compare across sample numbers. And the other key thing here was the slope of these lines was significantly different. So even comparing across, comparing across different sample numbers is challenging as well. Unlike Schmidt, what we found was autosomal heterozygosity is not robust to sample size in all cases. The really interesting thing, we specifically included the grasshopper K-gris because they found none of these things had an impact. And so we redid it to compare to our data sets where we're finding it, and we found it didn't have an impact. Sample size didn't have a huge impact on autosomal heterozygosity in this grasshopper. So they weren't wrong. It's just the grasshopper was weird. And I think this is because the grasshopper is a declining species. And so adding more individuals didn't lead to like polyallelic things. Whereas this eucalypt is quite widespread, has a really quite large population size. And in particular, the Kimberley rubella, which is the tree frog that you find in the toilets, the brown one across Australia. In the Kimberley, it is mega diverse. This is not a threatened species. If you want to find a non-threatened species in Australia, this frog is definitely one of the least. And interestingly, when we looked at Central Australians, so this is the middle of the desert frogs, and they have a lot less diversity and a lot less polyallelic sites, and they're more like the grasshopper. So your sample size effects on estimates of diversity are made worse by in your more diverse species. So that gets really confusing. What that leads to is a misranking of populations. So this is what we would say are probably reflecting the real level of diversity in the 3 species and their populations. And what you can see for the grasshopper, the order of diversity is correct. But when we look at the eucalyptus and this is its relative heterozygosity, what you can see is while for all of these, so basically the eucalypt was all the same, but when we used all of the individuals or use a lot of individuals, even though it was equal, it changed the variation. And that's because there was different numbers of polyallelic sites in the data. And for the rubella, when we used all the available samples, which were not equally distributed and very different, so 41, 21, and 19 samples, we actually got the wrong order. We got our southwest population being the most diverse, but it's clearly not the most diverse. And this was because the Kimberley was so diverse that we lost all the sites that became polyallelic. And so we have to be really careful and play with our data to make sure and use our knowledge. Like when I looked at the thing that made this paper was when I initially did SNP-based heterozygosity on rubella, it said that Central Australia, in the middle of a desert for a frog, was the most diverse population. And that's what led to this paper. Was actually, that makes no sense. So you need to remember your biology. So more individuals means, first, more invariant sites are added versus variant sites. And we added more individuals with more than 2 nucleotides at a location, resulting in this impact being highest on our most diverse populations. And so that's published 2 years ago. And so, yes, definitely use autosomal heterozygosity if you don't have a genome. Make sure you keep your rare alleles, because rare alleles are what drive diversity. So your filtering for population structure should be different from your filtering for diversity analyses. And you need consistent sample sizes, and you should do replicants and subsampling, and make sure you're testing the impact on your call filters when you're doing that. The next future of diversity is going to be, so this is the JEDI pipeline, which involves a number of people in the room, including Diana. So I used this for the anti-kinase argentis paper, and Shengyao's used it for the Davies A paper. So this gives real estimates of genetic diversity or of heterozygosity. It's great, but it requires a reference genome. And I think I might have been the first person to use and break this pipeline. And thank you, Diana, and everyone for helping with that. But I hit a huge problem, and that was because the species I was working on has an okay genome, but it wasn't good enough for this whole pipeline. So you need an actual pretty good genome. So it worked with a better antikinase genome, even though it was a different species. So you need a pretty good reference genome for that. But this is totally outside of R, and you'll need to run some command line programs, but it gets you unbiased estimates that don't lose all your polyallelic sites. The future, so this is, I think, where we're mostly at the moment is this building, and I'm kind of proud that most of this work has been done in Australia. Actually, all these papers are Australian. So the future things that we need to resolve is getting real estimates of DDRAD without a genome. So hopefully the next evolution of this JEDI pipeline is JEDI without a genome. Diana, please. Making sure that, which is essentially considering all the sites, which are polyallelic sites plus our invariant sites. Until then, so this is not a, here's your pipeline, run it, and you'll get an answer. You need to carefully think about how you design your study and understand how those choices affect your loci availability. If you create a catalog of loci, make sure you're not biasing it towards particular populations, and understand how that can affect your downstream results. And make sure when you're SNP calling that you are thinking about how that biallelic nature of the data shifts the information that you get across different sampling regimes, and know that your sample number will also affect that. So doing these tests does involve dartR. I use dartR pretty much every day, but it does require you to call SNPs multiple times. And it's hard for me to see these days a paper that gets through review with me where they've called SNPs once across their paper, and that made sense. So a really key thing is one set of filters does not fit all analyses. You will require different filters for the same data for different analyses, and that requires you to understand the theoretical background of each of the analyses that you are doing. I can strongly recommend Matt Hahn's Molecular Population Genetics is kind of written at a grad student level. Here's the theory you need to know to do good population genetics. I got our UWA library to buy it, and it basically lives in our lab. So some examples of things you would filter differently. For population structure, phylogenetics, you would remove your secondary SNPs and do LD pruning because you don't want linked loci. That's fundamentally incorrect for phylogenetics, because you want each of your loci to tell you a unique, independent story of evolution if you're using things like species tree approaches. But you might as well filter out your minor alleles, and somewhat hard too, because they just add noise to phylogenetic analyses. But if you're doing diversity analyses, there's no point, you shouldn't be getting rid of your secondary loci, because heterozygosity is heterozygosity. Private alleles are private alleles. If you're trying to represent the total diversity differences between two populations, why would you throw diversity away? And then demographic analyses are very sensitive to rare alleles. And you need to be very careful about your minor allele frequency filters, because if you throw away all your minor alleles, you immediately push your population into a bottleneck or one of the other scenarios. And so you can't break those filters away from the theory of the analyses and the tests that you're doing. So what I've put together for the rest of the session is, first, I put a couple tutorials up. So this is Galaxy Australia. If you're in Australia, you can log in through your AARF and you get lots of storage and data. But first is just a tutorial to Galaxy and how to use it. So Galaxy Australia is basically a big bioinformatics engine running off supercomputers. And when you log in, it's already installed like all of Stacks and everything you need to do all of your quality control from raw data through to SNP-calling. And in particular, the Stacks pipeline. And it removes that barrier of I need to install all of these huge command line things. So this first one is just an intro to Galaxy. It's not like the world's most instinctive interface, but at least it's a point and click. And then they have another one written by Australian Biocommons here, and that's actually how to run Stacks in Galaxy. So if you're like, oh no, this is how you can do it yourself. And as I said, none of the commercial things are bad if you have told them to do the right thing. If you have not told them to do the right thing, you either need to get them to call it again or you need to do it yourself, but you need to figure out your study design first. So if you want to, when we go to breakouts, there's an option to just start going through those tutorials and try and get your head around Galaxy as a way to run stuff. The other option is to go through your supercompute within your university. So things like Kaya here at UWA, I know that also has lots of software installed. So if you want to do that, also, you need to understand the Stacks pipeline. And I suggest Stacks not because it's better than everything else, but because it has the best manual. And the Google group is really good. And there's a lot of documentation on how to finally control your individuals in the catalog building. So the C-Stack stage is super important. You don't want to just run C-Stacks and then shove on all of your individuals. You want to build that catalog of loci, thinking about sample and study design. So it is slower than GATK and IPyrad, but it's just better documented and has the Julian Catullo actually replies on the Google group all the time. And it produces a VCF file that needs to go through a set of steps before turning it into a GenLite object where you can use dartR for lots of things. A VCF does have an advantage over a GenLite because it has finer scale data about loci that lets you do more thoughtful quality filtering. So things like read depth you can do at a different scale. So you can do it by individual by loci instead of an average across all of your loci. So for this session, the options are so I'm not going to pull up the POSIT cloud and run through it because I think you guys have got that. And from watching everyone here at WA, nobody's really listening. They're just trying to figure it out themselves. So I'm going to suggest we go to the breakout rooms now, and you can choose either to do the introduction to Galaxy Australia. And even if you aren't in Australia, that tutorial for Stacks is really well written. And I think the EU and lots of places have Galaxy available through your own organization, but that tutorial both tutorials will be helpful for you understanding how to do things in Galaxy and how to run stacks. Or I've provided a VCF file on a little bit of the Uberllia-Davies data set, and so you can work through that. So the purpose of that pipeline is not to do all the filtering, but to do that basic filtering to get you through to, I have got a well-specified GenLite object where I have thrown away the absolute rubbish. And now that I have that, I can choose how to filter it from my downstream pipeline. So do I want to take this through structure, population genetics, diversity? So the purpose of that pipeline is throw away bad loci, throw away bad individuals, check your duplication, check your reproducibility. So reproducibility is based on your duplicates, remove your duplicates, and then after that, that object is what you would use, that GenLite object is what you would use to filter for your specific data. And I'll make this PDF available somewhere, but I've just thrown in a whole bunch of links that you should read. These are all just methodological papers for population genetics. That are really useful for just thinking about study design and the practicalities of that work. And that's me.
Thanks. Renee, please stop me if that's wrong, but I think I make a poll and I kind of help what people can do. Oh, that's the wrong one, sorry. Give me a sec. This one. Is that was your suggestion? Is it correct?
Yeah, go into one of those.
yeah, and obviously you don't have to. You can do whatever you like. It's not like you have to go with the majority. I quickly show how you get there. Maybe that's one bit. The easiest way to follow the link if you want to have the Galaxy, but they're also on the website. And I share. Can you unshare your screen?
I thought I did. Oh, we're in the poll. Sorry, let me unpoll you.
Maybe it's my poll thing.
How do I unpoll you? Close poll? Nope, it's not.
Yeah.
Okay.
Yeah. Okay. Cool.
So do you want me to answer a couple questions?
Yeah, definitely we can do that as well. Before we go, people can obviously go in the backup groups and listen to your answers. It's beautiful.
Yeah.
So on the website, you find basically under tutorial 5 the links to the tutorials. Yeah, directly if you want to go there, or also on the website you have also the code, and that's also on the POSIT Cloud under tutorial number 5. So we could go there as well. And go to tutorial number 5 and find that. I just fire it up. Give me a second. So that's the two things. And yeah, Renee, you can also answer questions while this is firing up.
Okay. Okay, so a couple questions. So how do you choose which individuals to drop out when reducing to have equal number? And statistically, the answer is you should bootstrap it. So you should do that analysis multiple times. So you should like randomly select 5 and then 6 or 5 and 10 and 15. And you should do that multiple times. You should randomly select multiple times to make sure you're not getting some weird effects. Because one of those things that could happen is you could be like, oh, it turns out I have related individuals. And if I accidentally select them, my diversity drops. So the answer is you should run those analyses multiple times with equal numbers, but different individuals to make sure you're not getting a weird bias in your data set. Yeah. Is there a threshold, any threshold that a minimum number of individuals should be sampled from each site? We've tested this sort of in non-publication ways a whole bunch of times, and my answer so far across frogs, mammals, and spiders is 6. But what you're trying to find is, if you remember that there's that first slide where it's showing diversity creating an asymptote. And you kind of want to get to that asymptote on all your populations. But my rule of thumb for my lab is 6. Okay. I will share the slides. I just finished them very recently. So what would be the advice when working with nomadic species where sampling location may not reflect individual's population of origin? I know Nurjana, but I would not, I guess I would question whether that's not the population of origin. If your species is widespread and wandering, I think that's their biology. It just means their range is really big, right? I don't have a good answer for that one, sorry. So should you filter at all for math if you are interested in exploring heterozygosity? That's a complicated answer that intersects with read depth. So if you have really good solid sequencing, so you have really high average read depth across lots of loci, if you're confident in your rare alleles, I don't see a reason to remove them when you're measuring diversity. They are real, they exist. But if you have kind of poorer sequencing coverage, so your depth of a lot of loci is below 15, I would be really cautious and think about removing those because you can't be confident. So you need to make that judgment of how confident you are in whether your like singletons and things are real. So that will depend on amount of sequencing coverage, which depends on genome size, which also depends on money. So I think you just have to judge how confident you are. What other questions? Yes, Bill, when I said autosomal heterozygosity, so the Schmidt et al. Paper defined autosomal heterozygosity as basically biallelic sites plus invariant sites. So introducing the invariant sites allows you, so that is a value that right now you can only kind of get out of stacks and things like that. That's not a number you can get out of dartR, but it does include invariant sites. Yeah, and yeah, that repeated question, how do you choose which individuals based on your unedited sampling? Do the same number multiple times and draw from your suite of samples to make sure you have that. How do you know the number of the asymptote for our data? The answer is you have to generate it by generating your samples over and over and over at different sizes and do that. I actually have, if everyone needs that, I have code that I've done that with Antikinus to look for that kind of things. I work with eukaryotic asexual populations. I have no advice for you. I'm so sorry. I think you need to talk to plant biologists. I can't, way too hard. I'm very sorry.
Maybe
So,
Renee, you can say which is the question you are answering, and we can put it on stage.
Yep. Sorry, I'm looking at Cameron's question. The last one, would uneven sampling have the same downstream effects for larger scale evolutionary phylogenetics projects with multiple species? My answer to that would be no, but I wouldn't use ddRad for it. So phylogenetic analyses use all information. They use all variant sites and invariant sites. They're polyallelic. They use nucleotide substitution models. So you're sampling this, it's not the same, and the maths, the actual fundamental theoretical maths between phylogenetics and this are different. So if you have a larger scale evolutionary or phylogenetic project with multiple species, and what you're trying to do is build a phylogenetic tree, and that's your primary outcome, I would say that ddRad is not the right approach for you. I think the most cost-effective thing you can do now for that sort of question is genome skimming, like whole genome sequencing and then pulling out BUSCO genes and UCEs to get a suite of loci to build a tree, and that's going to be robust to sample size and be a lot more theoretically valid. And so this question from Jacob on when receiving SNPs called by a commercial company across a diverse, widespread species with unknown or unclear genetic structure, should I be calling SNPs again? And the answer is yes, you should be. And I know that like diversity arrays, I think they even say online, they will call 2 more data sets for you if you do get to them quickly. So if you get your data set from them, run it through pipelines and make those sampling decisions. As long as you do it relatively quickly, I think they have something online, somebody at diversity arrays can say, but they'll do more for you. I don't think AGRF will, but if you want to be a population geneticist in this world, you still have to learn to run your own things. The data they gave you and the raw data, like every time you order, you should get your raw data. And quite frankly, I now reject papers purely because people put up the SNP data sets as data availability. I won't accept a paper as a reviewer where you've not put the raw data on the sequence read archive. So you need that raw data anyways to publish your paper. You got to learn to do PopGen from raw data. Sorry, I have to tell you that. But the data like diversity arrays and AGRF generate is great. I only use those 2 companies to generate my data. And you need your raw data anyway, so do that. Sorry, I'll answer this one last question to be done, which is the anonymous. Does this kind of filtering, even sampling, SNP calling have to occur before sequencing? The answer is no. You can sequence everything you've got, but you need to do the SNP calling again. So you have 150 individuals, you then subset that raw data down to those 150 individuals and call SNPs on the ones that meet your study design. Okay, I will let it go on. Sorry.
Okay, so should we go now to the breakout rooms?
Yes.
Okay, here we go. Hello, everyone.
Did you pull us all back out? What happened?
Yes, I'm sorry. You are muted. Baron?
Sorry, it's me. I want to show one bit, and then you go back into the breakout rooms, because there's one bit information that's missing, because we just got the data yesterday, and it's all my fault, I guess. Doesn't matter. To get to the data to run the tutorial, please go to tutorial number 5 in the POSIT cloud if you want to do that. Yeah. And what you need to do there is you have to find the files, Renee said something, set working directory, find your files, and then you can load that. So the data files are under your tutorial, under data. Yeah. So if you go to tutorial number 5, maybe go really back. That's what you see normally. Go to inst, tutorials, number 5, and in data, there are the data. Yeah. And if you have this code, maybe I copied in the chat, I don't know, then you can access the files to run the tutorials. Otherwise, you probably will never find them. Yeah. There's a third file you have to find. That's the tech reps, which is coming later. And the path obviously is the same. It's just change this one with tech reps. Okay. So the file for the input, the folder, I put it into the chat. It's this one. So you should be able to run it. Sorry, and you go back to the breakout groups.
Hello, everyone.
Hey, Luis. You or me?
You.
Okay, I guess we are towards the end of the session. There was one question I know, but Renee can answer that. Is Galaxy available for people outside of Australia for free as well?
Australian Galaxy is not, as far as I know, but I know there's global implementations of Galaxy. So like I, go to Galaxy Australia, but I know there's a European Galaxy. I think there's ones in the Americas because, so the framework is global, but the compute behind it is local. And so you should investigate. I know there's a EU Galaxy because honestly, I use their tutorials all the time to figure out how to do stuff. So yeah, I don't know, but for Galaxy here.